what confidence do we have that this will actually work? I mean, have we tried it in animals? You talked about reanimating nematode worms.
Well, we can now move into chapter 2.
Okay.
Okay. So this is because your question is just bringing me to chapter 2. So what is the possibility, the real possibility of cryopreserving an organism, a biological organism, and reviving it? So there are many possibilities, many examples that this is possible. First of all, we can choose, but let's take artificial manipulation. And the more impressive of those manipulations, not the only ones, are embryos. The process is like this. A woman, is not very fertile, so the woman is induced to ovulate, to produce several oocytes, not only one, and the physician collects those, let's say just for the sake of an example, 10 oocytes. Then they have the sperm of the partner, and so they mix the oocytes with the sperm. Of course, the efficiency would be usually 100% because there is no obstacle. So probably the 10 oocytes will be fertilized, and so you will have 10 embryos. Of course, you cannot implant 10 embryos in a woman because that would kill her. So what you do is just, or they do, is save none. Just this is an example. Save none. They vitrify. They just immerse them very quickly, and that's the key element. Very, very quickly, instantly, so to speak, because this is a very small thing. So when you just drop it into nitrogen, it will freeze instantly because it's just a very microscopic thing. So it will take no time essentially for the cold, the low temperature to get everywhere inside that. The result of this, if you can imagine the molecules there, is that the molecules in the cells are everywhere in chemistry are moving all the time. So when you freeze them so rapidly, they don't have any time to do anything, to reorganize in any way. So it's like a snapshot. So the molecules were in a certain situation, and they are frozen, and they remain in that situation. Then you thaw that embryo very fast, very, very fast. Again, they don't have any time to reorganize, move to a different position. They suddenly wake up, and they are in the same position they were before. So they keep living because nothing happened to this molecule. If you have a human or a mouse or a rat, a brain, so to speak, you cannot do technically, so far it's not possible to do that. It will take some time. And when the temperature is just going down progressively, even if it is relatively fast, but if it goes down progressively, molecules, while they begin to get cool, they reorganize, so they tend to be concentrated in the center of the fluid, and the periphery is more diluted, like water. So this changes everything. At the end of the day, when you finish the process, the molecules have reorganized. That you cannot avoid that because this is chemistry. Then when you thaw that body, you cannot do that instantly. In any case, they are already reorganized. But you have to do this progressively because there is no other chemical solution. And so, again, the molecules thaw progressively. They keep disorganized, might just disorganize further. So at the end of the day, when that body, that brain, is completely thawed, it is no longer the same that it was cryopreserved initially, because all the molecules in the cells have changed, have got disorganized, and this cannot be undone, at least now. So we cannot, so we have to we know how to take off, so to speak, but we don't know how to land the ship, the plane. So we have to wait for the future to achieve that. So this is why it is not certain, but of course, as I said before, if you go to a grave, you have a certainty, 100% certainty that you you will end up in the stomachs of the worms, and all your molecules will be spread, and that's it. That's it. In the other case, you have a tiny possibility so far to be realistic, but it's higher than zero. So as I said before, you have really nothing to lose. If you are rich or have enough money and life is sufficiently worthwhile for you, you want a second chance at life. It may fail. Well, in any case, life is a risky activity, even for a baby, for everybody. So we can just get struck by any kind of injury and die immediately. So we are very fragile, and this is another instance of risk that we are taking. But if we hang on to life, well, we do our best to remain alive. As I usually say, or some people, and I say that for the individual, death is a tragedy. For the species, maybe not. It's a good thing. But for the individual, because there will be no other Richard, no other Rudy, because our molecules will never come again as they came again one time. Just consider, for example, the first stage, the fertilization. You have your one oocyte, but millions of sperm cells. So it's just a kind of a miracle that cell, that spermatocyte, just fertilized that egg. If we try that several times, millions of times, probably other spermatocytes will just fertilize that egg. So, and this is just one example of how many casual events, many coincidences happened at random, and this will not be repeated. So this is why this is our only chance to be as we are, and this is why we want to preserve the cryonicists. Of course, the public in general, because there is a tiny cryonics community enlarging. That's true, but it's still tiny. Think, well, this is a chance, very slim. Yeah, that's it. So perhaps it is not. Many people may think that it is not worthwhile the effort and the expense they have to go to just for such a small chance. This is a way to think. To think. But as technology improves, and that this is happening very fast, chances are getting higher. And if we come back to the embryo, that embryo, let's say a human embryo, so they saw one embryo very, 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 very fast, as I said. So this embryo usually is viable if the technology is okay, and so it is reanimated, so to speak, and in fact that's a human being in a very initial stages, but all potential possibilities are there. So if you screw up with that just a little bit with the embryo, the baby will be born with defects. So, but obviously the embryo recovers perfectly well. The physician implants that into the mother, the uterus, and maybe the second or third or fourth time, the woman gets pregnant. And there are thousands of people that started their life now as a cryopreserved embryos, and they are alive. Apparently nothing happened with their soul, considering religious people. So that is quite feasible, because these people are just quite normal, you know. The same can be done with animals, of course, and this is routinely done now. So this is a technique, well, with certain degree of sophistication, but now it's relatively routine for people that are experts in that. So this is one example. Also, this is worthwhile mentioning. There is another possibility, always provided you are taking a very small sample of a tissue. Let's consider ovaries in a young woman, so a 25-year-old woman, and she has the misfortune to get cancer. So she will have to undergo chemotherapy, and that will probably kill all her reserve of oocytes, because a woman, an ovary has a lot of immature oocytes, and they just use one every month. The others some of the others disappear spontaneously. So the initial number may be when a baby is born, maybe about 300, 400, something like that. Anyway, it's a limited number. So with that therapy, all the reserve of immature oocyte that this woman has is killed. So that woman will enter into menopause immediately because the ovaries would stop working properly because they lost the important component, the oocyte. And then, of course, she will be unfertile, unable to get pregnant. So what is the solution for that? Well, the physicians in Japan, for example, I know that they do that. It's not a difficult technique now. They take just by laparoscopy very small biopsies of the cortex of the ovaries. That's the place where the oocytes, immature oocytes, are stored. So it's relatively simple. So they take very small slices, and they freeze that instantly in the way that I explained to you, like an embryo, or using also a vitrification solution. And then they save those biopsies, a number, I don't know how many exactly. So let's assume that that woman is treated with chemotherapy. Everything goes okay, which frequently happens. And so she's cured of that disease. But of course, she's barren now. So what the physician does is just implant, because there will be no rejection because it is tissue that belongs to the woman. They reimplant into the ovaries a few of those biopsies, and that tissue will regrow. Of course, this has its allotment of oocyte, immature oocyte. So that woman, and this is really what happens, began to cycle again and to ovulate again, at least for some time. And of course, and they terminate menopause. They restart being fertile and having menstrual cycles and everything else. So this is a very useful application of cryopreservation. You may think of cryonics into cell tissues. Well, so this is the example that the technology, which is current now, is not a new technology, is able to achieve. And this shows that really tissues and full organisms can be cryopreserved and restored to life without consequences. Of course, what is the reason why that cannot be done with a baby, for example? Because we are too big. So the technology, the process is possible. What we don't have yet is that mature technology, mature enough to be able to freeze and thaw large organs instantly, because that is a condition, instantly. But this is a problem that we have now. But technology, you can be certain, will overcome those limitations in the future. It's just a matter of waiting, and perhaps waiting less time that we think that we perhaps would wait. So this is not a problem of if it is possible. It is a problem. A question is when it will become possible. But it is definitely possible. And so, if we have the time, I can now give examples in nature, because this is not new in nature. And I will take just two examples. Also, small animals, but with a physiology which is different for us. They are simple animals, but they have a tardigrades. This is a very small animal. The water bears, also, they live usually in humid places, and they can, if the animal lives in Siberia, for example, where the winter comes, temperatures go down to -70 perhaps, or -50 Celsius, so they freeze. They have certain adaptations, so they freeze. They have a simple, but they have a nervous system with neurons like ours, exactly, that is less complex. But they can even learn things. The other example is the worm C. elegans, which is also small. So those can be, well, in nature they freeze not instantly, they freeze progressively because winter comes little by little. But they are adapted to that, so they freeze, and that is the tardigrade. Perhaps the C. elegans that live in Siberia can also freeze. I don't know that for sure, but they can resist that procedure. So when spring comes, they thaw, and they keep on with their lives and reproduce and live happily. So this is an example Here, it doesn't matter much that the animal is small or not, because the instant cryopreservation is not used by nature. But these animals are adapted after probably millions of years, and so they tolerate this because they produce probably, we don't know, at least I don't know how they manage to do this, but they do. And so this is a very excellent model system. NASA is using those animals for other purposes, but also because they are called extremophiles. They can tolerate very extreme condition of heat or cold and survive. The price that you pay to be such a resistant animal is that your brain will be simple, and your body structure cannot be sophisticated, because if you have a sophisticated brain, you need a lot of protection for your brain, because that brain is, and usually our brains, complex brains, do not tolerate that. They are not adapted to this kind of changes. So simple animals can tolerate this, but the price they pay that they have to be simple to tolerate these changes. Sophisticated animals like mammals cannot tolerate that. So there is no example of that in any kind of mammal. This is definitely, at least to my knowledge, which I think is reasonably complete concerning mammals. But this is a process that can be done. And speaking of other animals, well, the C. elegans, that worm that is very common, can be cryopreserved by the same method that I described to you for embryos, and they can be reanimated. And there was a very interesting experiment that was done actually initially in Seville. There's a man, a physicist there. His name is Ramon Risco, is in Seville. And so he works with worms. So what he did, and then it was repeated and published in Alcor, he cryopreserved these worms. But before cryopreserving them, they can learn. They are very simple, but they have a brain and can learn very simple tasks. So they can be trained. So he trained those worms, C. elegans. Then he cryopreserved them, reanimated them, and then checked if the animals remember that training that they learned. And they remember perfectly well. Of course, you may say, well, these were simple tasks. But anyway, the nervous system works in the same way for us and for them, except that we are more complex and they are simpler. But if it works in them, there should be a way to make it work in us. Of course, this will involve a certain level of technology, or perhaps a high level of technology. But again, this is possible. This is not an impossibility, some biological impossibility. No, it's not. So that was a really very seminal study that was published. And it really is a quite encouraging study, because it shows that in a simple animal, but anyway, an animal with a brain, memories can be preserved after cryopreservation in the same way that we use for embryos. Well, embryos is different because they are not trained, but these animals are really adults. They are trained, they learn, and they remember perfectly well. So in cryonics, I am one of the proponents that cryonic research begin to use these animals to begin to study and to develop the techniques of reanimation. Because now we leave all the responsibility to the future. But the future must start at a certain point. Well, why not now, I say? The only person that does that in the world is that man, Ramon Risco. And well, I know him, of course, and it's quite important what he does, but that he should be helped to develop a research line with that in cryonics, because now cryonics is growing, and so it is reasonable to think that we have to also grow in terms of experimental activities. It's like biomedical sciences in general. So cardiology, of course, takes care of patients, but also there is a lot of studies, basic studies in rats, in other animals, concerning cardiology and cardiological pathologies. Well, the same thing is valid for cryonics. So there should be experimental work trying to reanimate or to understand how these animals, what is their brain that allow them to be reanimated and to preserve their memories. And there are many tests that can be done that will be little by little constructing the knowledge and the technology for to be able one day to begin to reanimate larger animals, not really a human initially, but larger animals little by little. And so is the way biological sciences or biomedical sciences evolve. This is a step that has not been taken by cryonics so far. Cryonics just keep doing the same thing that it began to do, perfected, but the same technique, cryopreserving the brain. But that's okay. That's okay. I have nothing against it. But new research has to emerge, basic research, because this is like any biomedical science need to use new research. So this is what I say. So far, I don't have much company in this, but at least this is what I think. But sooner or later, I think that someone will begin to, beside this man, to do this kind of experiments. And what is more important, to get funding to increase the complexity of those experiments. So this is what technology has been done so far, Richard. So, of course, this is not a final proof, but gives hopes and shows that it is definitely possible biologically, naturally and artificially.
Right. So you did the process, or you have done the process on rats, right? And then you thawed them out, and then you looked at the brain. Did you learn anything? What did you learn from that? And did the brain look okay?
Yeah. Morphologically, it looked okay. They were well cryopreserved. At morphological level, this is one level. I didn't study the biochemistry of those brains, let alone the physiology. So the only thing that I can tell you that the cells were, and the structure, the histological structure of the brains were in general well preserved. Some cells were less well preserved than others. It's some type of neurons, because we use a technique that is called immunohistochemistry that identify different kind of cells of neurons, because we have different kinds. Some types of neurons were better preserved than others. But so this is what I can tell you. So it was not a perfect cryopreservation, structural preservation for all the cells, but on the average they were well preserved. So if you ask me, they can be revived? I have no idea, and I can make no prediction. This is what that experience that I had with rats made me think that, no, there are many good people doing this kind of thing. I don't want to do this thing because I don't feel satisfied. I want to reanimate. I want to use an animal that can be revived. And for that, I have to go down to worms. And I'm willing to do this. I have convinced one of my students to do this, to go and spend some time with Ramon Risco in Seville to learn that we are very close. He's a very generous man, and probably in a year or so, perhaps I will be able to say, well, Richard, we have begun in Argentina to do this kind of work, just modestly. But one advantage of this research is that it's inexpensive in general, because worms you don't need an animal facility for that. These are like bacteria. You have just an incubator, and that's it essentially. Actually, in my institute, they work on different biochemical areas. One is lipids, and they use one group uses C. elegans, that is some lipid metabolism. So because C. elegans is very widely used in biology, and so this is an advantage. So this is my plan concerning cryonics. I am not fully involved. I am not planning to be fully involved in cryonic research because I have limited time. So my full time will be devoted really, and the time of my group, on rejuvenation. But I am a cryonicist. I am not the only one in the lab. My students don't care at all about cryonics, but they find an interest in cryobiology. And also, of course, it's important that they can publish, because this is a vacant niche. So if you do things, probably will be one of the few, perhaps the only person doing this kind of research. And there are journals that are friendly to this kind of research. because now cryonics is more accepted than in the past. This is a good thing. And in Argentina, nobody has any problem with cryonics. So that is an advantage. It's a quite tolerant um, country in this situation. So there are no religious problems. We have enough problems of different types, economic, especially. So to be concerned about cryonics, which is
Right.
an advantage.
Okay. Okay. So thank you. I think that gives a really good understanding of kind of where we are with cryonics. So that's really interesting. And the talk on cryogenics is a great introduction to the technology. So I think that will be really useful. And
So, and it's the only, for people old, there may be no time to wait until, for example, rejuvenation is achieved by medical techniques. So we can take a chance, which is cryonics. This is not a solution of the problem. It's a waiting room. So you can go there, enter that room, and wait. You don't know whether you will be if you will wake up or not, but at least you have some hope, and you know that it is possible having the right technology, and so this is a possibility against nothing. So say 50 years ago or 60 years ago, this possibility was not available, and now it's available, and so we have at least an option for some people. And the number of people signing up for cryopreservation is increasing, really. The number of organizations, the biggest player now is China, because the Chinese have the support of the government. This is very important. So whereas in the Western world, in the States or Europe, they are just tolerated currency, but they are not supported. So the Chinese have a big advantage. Their government, apparently officials, have a vision which is farther than the Western leaders.
Yes. Yeah. Interesting. I mean, can I just ask, are you planning to try and have
I have
yourself
signed up. I
you're
have
signed
signed up. My problem is that I live in a faraway country, and I am not planning to leave Argentina because I feel comfortable here. So transportation is the problem. Well, there are many problems even within the states. So what we have a small cryonics group in Argentina, and as I mentioned just in passing, we have cryopreserved a human brain in Argentina. Was really I was not involved because I was just out of the country at that time, but I was small group just with the help of a physician and a funeral director with experience in this. Well, perfused and extracted the brain of that patient that was a cryogenesis too, a relatively old woman with an incurable disease. And the brain was sent to the States to be stored there. So that was an achievement with with a number of limitations, because it was our first time. We were not trained or well equipped by. We gathered information from other cryogenicists around the world, and so with all that information, we could just do that. And so that was the first experience. But of course, like in every activity in science and in other areas of human activity, money is essential. And we are facing the problem of funding in Argentina and perhaps in general, because as NIH will not fund a cryonics project, not at all. So there is only one foundation. I mean, there are 3 or 4 foundations in the States, only in the States, supported charities, supported by Bill Falloon is his name. He's the man that supports the cryonics that is being done, the cryonics research in the States. And nowhere else except China, perhaps, there are no charities, as far as I know, supporting cryonics research so far. So this is really what also is keeping cryonics from taking off. It should. But anyway, it is the number of people has grown, the number of people signing that. So the confidence in cryonics is higher, And as technology improvements are observed by people, they are more confident in the possibility that really a second chance. So little by little, that is making progress too.
Excellent. Thank you all for watching. I hope that you found the video informative. Please do hit the thumbs up button, subscribe to our channel and hit the bell button for new video release notifications. It encourages us to continue to create more videos about anti-aging and extending healthy lifespan. Thank you so much for your kind support. I wish you all well and will speak to you again soon.